Hi, I'm Dr. Joseph Gleeson, Chief Medical Officer for the n-Lorem Foundation. This presentation is to help physicians submit nominations to the n-Lorem Foundation for personalized antisense drugs. Thank you very much for your interest in n-Lorem, and we're excited to share this information with you to help make your submission most likely to be successful. The mission of n-Lorem is to apply the efficiency, versatility, and specificity of antisense technology to charitably provide experimental antisense oligonucleotide medicines to treat patients with nano-rare disease, meaning less than 30 patients that carry that mutation in the United States. Submissions as of September 2022, 158 total submissions received, and of those, we've accepted 78 cases. There's 53 that were declined for various reasons, and the purpose of this session is to improve the chances of your case being accepted. The 74 cases that were accepted are in progress now to have drugs developed. There are 9 that are on hold pending additional scientific information. There were 4 cases that were initially accepted and then later declined because either we were unsuccessful in developing a drug, or in one case, the clinical course was too far advanced to expect much benefit. There's several routes for administration of antisense drugs. Systemic can include intravenous, intramuscular, and subcutaneous. Local can include intravitreal, intrathecal, and aerosol. And it's important to consider the site of the disease cells and how to get antisense drug into them by administering the drug to the right compartment. The process of developing a drug starts with the identification of patients along with their genetic information that would predict they would benefit from an antisense drug. Generally, these are at tertiary care medical centers with physician scientists caring for patients with a good understanding of genomic causes of disease. They then submit a nomination to the n-Lorem website, where we take that nomination and evaluate it for the likelihood of success. There's often back and forth with the physician, several questions, and to determine the appropriateness of the patient for n-Lorem. We then finalize the case for presentation to the Access to Treat Committee, ATTC, which is a group of peer physicians that have experience with genomics and antisense drugs to assure the patient's appropriate for the n-Lorem approach. Once approved, the program then goes into the drug development phase, where literally thousands of antisense drugs are screened for the genetic mutation or the gene therapy approach to develop an optimal antisense drug. Those drugs are deemed to be safe through a series of safety measurements. And finally, the drug is produced at scale through a sterile fill and finish, and then sent to the prescribing physician. In parallel, the drug is submitted to the FDA through an investigator-initiated IND submission based off the FDA guidance for N-of-1 antisense drugs. Finally, the drug is administered to the patient. Then n-Lorem works closely with the physician to assess potential side effects as well as potential achievement of primary and secondary clinical endpoints. What makes n-Lorem possible to provide drug to patients for free for life is that antisense drugs generally are not metabolized by the body in a natural way. So they last in the body for several months in general. So a very small dose can last a long time. Intrathecal dosing for CNS disease, generally administered every 3 to 4 months, with a very small dose, 500 milligrams, and it can permeate the entire brain and can restore cellular health. Similarly, other organs, relatively small amount of drug. So with one synthesis, we can manufacture enough drug for a patient for 10 years. And this makes the manufacturing costs quite feasible for us. I'd like to review some of the characteristics of a patient or a mutation that make it not amenable for n-Lorem. First would be if the patient does not have a single gene disease. If there's multiple genetic variants or multiple single genes, not appropriate. If there's unclear genotype-phenotype correlation or not enough known about the gene or the patient's natural history to make a clear decision. If the mutation is a total loss of function due to a frame shift or loss of a splice site or ambiguous mechanisms of action, we can't proceed. On the other hand, if it's a splice gain mutation, or there's ways of manipulating gene expression with antisense, then these should be carefully considered. If the disease is too severe or too far advanced to measure benefit following a 12 to 18-month treatment protocol, if the patient is treated exclusively by physicians outside of the US, if the disease affects organs not currently characterized or amenable to antisense therapy, if the physician and institution are not capable of treating the patient for the long term, up to the entire life of the patient, or if the disease is caused solely by developmental or structural mechanisms. The basic information that's required for submission to n-Lorem include the physician and institutional readiness, a very clear patient evaluation, clear genotype-phenotype correlations, and the more information that's available, the better. Greater clinical and genetic information can speed the process. And we encourage you to contact n-Lorem with questions during the submission process. The mechanisms of the antisense drugs that n-Lorem is working with are targeted to several different potential approaches. First, toxic gain-of-function mutations are the #1 indication. These are mutations in genes that lead the protein to malfunction in some way and impair cellular health. Antisense drugs can knock down a specific pre-RNA haplotype and render that gene silent and keep the healthy copy active. Partial loss of function mutations, such as a gain of splice site, if there are ways of upregulating genes by taking advantage of things like poison exons, we can consider that. We haven't yet accepted any genes where there are total loss of function mutations for the reasons that I mentioned. And there are several nominations we've received where additional work is required before accepting the case. It's very important that the physician and institution are ready for the drug. And we consider several aspects in that decision. First, does the patient live in the US? Is the physician and institution capable of treating and monitoring the patient, both in the short term and in the long term? What's the institution's ability to financially support the trial and beyond the trial? The trial lasts for roughly a year. We expect that there is going to be some costs to the institution for the IRB, for safety monitoring, for assessing outcome, and for the physician's time and the infusion center's costs. We consider what experience does the physician and the institution have with treating patients or conducting investigator-initiated IRB-approved INDs, and does the physician or institution have experience with regulatory agencies? In the patient evaluation, it's very important we have a very complete picture of that patient and how that patient fits relative to other patients with that same gene mutation. What is the patient's history, the course, and the clinical status? What outcome is expected from the treatment? How severe is the disease? Is it likely to be stable during the 18 months that it takes to develop a drug? Are there fixed deformities or structural malformations? Is the proposed target gene expressed in the relevant cell type? And are there organs affected beyond the target organ? And are there relevant patient cells available for proof of concept experiments? When considering the genotype, it's important to consider the presumed function of the gene, the presumed function of the mutation, the zygosity of the mutation, is it heterozygous or homozygous, and the likely pathogenicity of the mutation as determined by ACMG guidelines. The effect of the mutations could be a loss of function or a gain of function. ACMG doesn't really carefully distinguish between these. They just look at pathogenicity, but in terms of antisense drugs, it's very important to distinguish between these possibilities. Toxic gain of function, loss of function mutations, whether they're complete or partial, and dominant negative effects need to be taken into account. Some of those aspects can be understood by mining data on the web, and some of it requires experimentation or collaboration with experts in the particular gene. Does the mutation affect gene expression? If so, that's another potential way that antisense drugs can work. So then we take the genotype and the phenotype together in some and look at the genotype-phenotype correlations in the evaluation. In other words, is there evidence for a single causative mutation? How similar are the clinical features to those reported in the literature? Is the gene expressed throughout life? Can an antisense, regulate the gene expression? Are there publications in animal models to confirm the pathogenicity, the signs and the symptoms? Are there publications in animal models that modulating the gene expression could show clinical benefit? And what's the on-target liability? In other words, if we can target the gene specifically, that's wonderful, but sometimes the antisense will work on the other copy of the gene or on off-target genes. And we have to take that into account, especially when considering the risks. So what are the consequences of the complete null or overexpression in model systems? In other words, what does the knockout mouse look like? Are there known human diseases with different zygosities, such as loss of function or gain of function in the causal gene? And is loss of gene function tolerated in humans? So to summarize, I hope this information is helpful as you craft your nomination. To summarize, it's very important to have a full clinical description and genotype presented in the nomination process, a careful consideration of clinical endpoints. What is it we hope to achieve? Assessment of the patient and the physician and the institutional readiness. These are long-term commitments we all are making to each other to ultimately help the patient. We need to consider cellular mechanisms and to consider the potential risk-benefit ratio and the timeline to therapy. Thank you very much for your attention, and we look forward to receiving your nomination.